what I'd like to do today is actually share with you a couple of stories. First of all, oops, yes, I have the wrong. This is my conflict of interest slide, and I guess the bigger thing is the IP for a lot of the work we've done is either at Rockefeller or more recently at Yale. As many of you know, our laboratory is primarily interested in two broad areas. One is immunobiology of tumor microenvironment in human cancer, and other is biology of dendritic cells in human immunity and inflammation. And I'm going to talk mostly about the first half of the laboratory's interests. And so just because it's a grant, I want to begin with a case presentation. This is actually an old story that began almost 10 years ago. 48-year-old gentleman who had, was diagnosed with multiple myeloma and had received what was conventional at that time called VAD chemotherapy followed by stem cell transplant, which was unsuccessful in the sense that he relapsed and then received several kinds of salvage regimens without benefit. And then was referred to us for evaluation of investigational therapies versus consideration of hospice. So you can imagine that the outcome for myeloma in those days was not very good. And we actually offered him participation in a new clinical trial that was testing a drug called thalidomide as an anti-angiogenic agent in myeloma. And lo and behold, he actually had a wonderful response to this drug and actually a durable response as shown here with reduction in the tumor-associated monoclonal protein. And then we actually went on to publish the entire experience, which at that time was in fact very difficult to convince the community that a drug that didn't really have an effect on tumor xenografts or a major effect on tumor cells in general could actually have a meaningful impact on cancer. And, but I think several thousand patients later, many phase 3 trials later, I think it's clear now that these drugs in fact do have a role to play. And in fact, the course of multiple myeloma has changed with the advent of three new classes of drugs, as shown in this slide, that all of them in fact either act primarily either on the microenvironment or specifically target the interactions between tumor cells and the microenvironment. This is not just true for the thalidomide, but also for its analog lenalidomide, as well as for bortezomib, which also has major impact, as we and others have shown, on the interactions between tumor cells and the microenvironment. And in fact, with the incorporation of these drugs into frontline therapies, you can see actually what we can see is that not only are we changing the survival of myeloma at population level, this is just the data from the Mayo Clinic, but also if you look at the most recent survival curves or the trends, if you will, from the current cooperative group trials that were actually having a significant impact on outcome in this disease, at least in terms of early mortality. So I think what the clinic has really taught us is that therapies that target the tumor microenvironment can have a significant impact on survival of patients with established cancer. And I'd like to make an argument to you today that perhaps it might sound heretical, but that specific manipulation of the tumor microenvironment may have an even greater impact on specific prevention of cancer in defined healthy individuals. So if you really want to think about prevention, I think this is the model we're going to work with, which is what we call basically a model for transformed plasma cells. So when the great majority of times, while clonal expansion of transformed plasma cells is a prerequisite for development of multiple myeloma, the great majority of times this occurs, the clinical outcome in fact is not myeloma but an entity called MGUS, which is a stable intramedullary expansion of transformed plasma cells. And it occurs in about 3 to 5% of people over 50 years of age, and only a small proportion actually transform to get clinical myeloma. But one of the surprises with the advent of interface cytogenetics and gene expression profiling in this disease has been the recognition that the tumor cells in MGUS are in fact a lot closer to myeloma than they are to normal plasma cells. And this has led to the hypothesis that perhaps the interaction between tumor cells and the microenvironment may be a key determinant of clinical malignancy in this process. And this is really a theme, I think, bearing out in many settings, but this is just, I think, we believe a model to really evaluate this process. Now, if you really want to think about prevention, I think people think of HPV-driven cancers as perhaps the gold standard. And if you think about the immune system, it is just useful to remember that the relative risk for getting or increased risk for myeloma in the immunosuppressed settings, such as transplantation or with HIV/AIDS, is in fact the same sort of risk, if you will, as for HPV-driven malignancies. So if you're thinking about trying to understand prevention of myeloma in this setting, the first question you want to ask is, does the immune system in fact recognize these so-called pre-neoplastic or clinically invisible lesions? And the pathologists have known for a long time that a lot of pre-neoplastic lesions have T cell infiltration. What they have not tried to do is try to understand what those T cells recognize. And so we've actually tried to, some time ago, we tried to ask the first question, which is, does the immune system recognize MGUS cells? And if you basically ask that question, the answer to that is yes, which is that if you look for specificity of T cells that are infiltrating MGUS, that's actually a very specific response that's specific for antigens derived from MGUS cells. And so the question is, can you go down to actually specific targets, and are those targets differentially targeted in diseases where the patients do well versus the patients don't do well? And so we began this approach by initially using a high throughput screen to ask for just a broad range of antigens that we could find. And the surprise there was that there were a set of antigens against which immune reactivity or the presence of immune response was associated with extremely good outcome. The top gene on that list, and the top 3 genes in that list are shown here. And the top gene was a gene called SOX2. The second gene is also very interesting, which is DNA methyltransferase 3-like. Now, this is just kind of validating that data at the level of reactivity for T cells to show you that if you, this is an assay where we take overlapping peptide libraries from these antigens across the whole protein and ask for expression of a chemokine called IP10. And you can see a production of IP10 in response to SOX2 in patients with MGUS, but you don't see that in patients with multiple myeloma. So just around that time, SOX2, in fact, initially was well known as a core factor for pluripotency and stemness in embryonal stem cells. But just around that time, Shinya Yamanaka published his pioneering work showing that this is also a part of the core factors required for what is called inducible pluripotency stem cells, for converting an adult cell into a pluripotent stem cell. It has also been, these sets of genes have also been now shown in putative so-called cancer stem cells in many, many settings. And there's a correlation between ES or pluripotent target signatures with poor outcome in several settings. In fact, it is now recognized that, at least in some cancers, such as squamous cell carcinoma of the lung, that these genes are in fact major oncogenes. And this is just data from Bob Weinberg's lab showing that that ES-like gene signature correlates with the outcome in, for example, breast cancer. You can do the exact same experiment in many, many human cancers just using the published microarray literature. And this is data for multiple myeloma, again showing that ES or ES target genes, again, correlate with outcome in multiple myeloma. The immune system, however, is actually very specific and in terms of being able to suppress or target these cells. And so if you, for example, if you take anti-SOX2 T cells, they actually have very profound effects on inhibiting the clonogenic growth of human MGUS cells. And this is shown here. If you basically stimulate T cells with SOX2 and add them back to a clonogenic assay, you actually have a dramatic effect on the growth of human myeloma, of human MGUS cells. To be able to ask what the clinical value of this was, we were able to take advantage of a study that we actually began some time ago to prospectively analyze and characterize this set of diseases through applying essentially a large number of omics-type approaches in a prospective fashion. And this study is now enrolled over 200 patients, and this is the data from the last cut that we did with over 100 patients enrolled. And you could go back and look at the presence of T cell response, correlate that with outcome, and you can see that the presence of SOX2 T cells seems to predict low probability of progression to clinical myeloma requiring therapy. And indeed, in the multivariate analysis, the presence of these T cell responses overrides all the other clinical variables that we currently use in the clinic today, such as percent of bone marrow plasma cells, level of M protein, and other variables. And the only two things that came out in this were anti-SOX2 T cells and elevated C-reactive protein. And I'll come back to CRP in a while because we think of this as a marker for inflammation or IL-6 signaling. So to summarize, expression of ES core genes or their targets seem to be associated with aggressive clinical course in patients with myeloma and many, many cancers now. And the presence of spontaneous T-cell immunity against SOX2, we believe, is an independent prognostic predictor of indolent course and seems to protect, or seems to be associated with reduced risk of transformation in myeloma. So the questions that I think we need to ask at the present time, and this is really work in progress, is to understand how do people lose that immune response, if you will, from MGUS to myeloma. What is the whole spectrum of the entire ES signature, if you will, that elicits an immune response in people, and are those responses seen in people like you and me? And that is something that we're beginning to answer with a yes, but that's something still a work in progress. What is the underlying biology of altered expression of ES genes in cancer in general, and specifically in multiple myeloma? So the finding that increase in IL-6 or signaling and or C-reactive protein also is a major predictor of outcome also, I think, brought us back to the old days of when we used to think about IL-6 in context of myeloma, and has really, I think, put inflammation back on the map as a major determinant, if you will, of progression. And this is really something that we've known about for quite some time. We've known about increase in inflammatory cytokines, TNF, IL-1, IL-6, bioactive lipids in this context of this disease. We also recently shown that there's an increase in a subset of cells called Th17 cells in the microenvironment in myeloma. We also have shown that the immune system, in fact, can recognize specifically inflammation-associated lipids. And that opens up a possibility for the inflammation to be actually controlled in a specific manner by targeting these molecules. I'm not going to talk about those stories, but talk a little bit more about the kind of cells that inflammation recruits. And one important cell type there is dendritic cells. The reason why it again goes back to a story almost 10 years ago when it was first described that myeloma lesions, in fact, were very enriched in dendritic cells. At that time, there was a bit of a controversy because the initial stories claimed that these cells were infected with a virus called KSHV, but that could never be reproduced. But in the process of refuting that observation, everybody was actually confirming that, in fact, DCs were actually enriched. And we forgot about this for several years until more recently, we and others have gone back and revisited the interaction between dendritic cells and myeloma cells to make two sets of observations. One, that the DCs seem to provide a major trophic and survival signal to myeloma, and that seems to be mediated by several factors such as BAFF or APRIL, rank ligand, and possibly some co-stimulation. But another important aspect, and this aspect I think is important from the concept of so-called cancer stem cells, is that the microenvironment-derived signals seem to have the capacity to alter the differentiation status of tumor cells. And so, for example, this interaction leads to the interaction between DCs and tumor cells can lead to a cell type that actually is more like a B cell in the sense that it actually starts to re-express molecules like BCL6, which have to be suppressed or extinguished before a B cell becomes a plasma cell. And in fact, more recently, the group at the Dana-Farber, in fact, went back and looked at the myeloma marrows to confirm that indeed BCL6 was present in the plasma cells of the marrows of myeloma, as opposed to what we would have originally predicted, thinking that these are typical plasma cells. The other thing that we observed in these experiments was that when we were culturing them together, that we could see a formation of multinucleate giant cells that was happening very quickly. And this was happening primarily when we cultured dendritic cells and myeloma cells together, not when we cultured macrophages or monocytes. And these cells, in fact, could eat bone, and if you put them in a bone slice, you could show their ability to eat bone. So what we were seeing was a very facile formation of osteoclasts. And so we decided to actually understand the mechanism of this as to what was happening. This is a cell contact-dependent process, and I'm not going to show you how we got to the mechanism, but to the mechanism, which is that this process requires an interaction between two molecules, CD47 expressed on myeloma cells, and the induction of thrombospondin 1 on dendritic cells. And you can knock down either side of the story. You can use an antibody that blocks the interaction between TSP1 and CD47. You take away the osteoclast formation. You knock down CD47 by RNAi, and you take away osteoclast formation. So this actually was very interesting to us because, indeed, bone disease is a major issue in human myeloma. But then, actually, this is also interesting to us from the perspective of the crosstalk between how cells interact with each other, because CD47 and is particularly been well implicated in as a do not eat me signal. And it appears that the same sort of signal nature implicates for a cell fusion event that leads to formation of osteoclasts. The question then was, if this is actually happening in the context of myeloma, is this actually very specific to myeloma, or can this occur in many other settings? And so we asked whether if this interaction is required for formation of osteoclasts from, let's say, rank ligand and MCSF, and this is a very standard methodology that people use to take human monocytes, culture them with rank ligand and MCSF to get osteoclasts. And indeed, if you block 47 TSP1 interaction in that process, you take away osteoclast formation from human monocytes. Now, does this happen in vivo? And the answer to that is also a yes. So this is a model wherein where you infuse, this is a collaboration with Carl and Sagna and colleagues, wherein you model where you infuse human parathyroid hormone into mice, and basically what that does in a rank ligand dependent process is cause hypercalcemia and bone loss. And indeed, if you actually inject the anti-TSP1 antibody before you inject parathyroid hormone, it takes away the hypercalcemia and preserves the bone in vivo. So I think what this I think suggests is that the 47 TSP1 axis is a broad regulator of skeletal homeostasis, but also that it is a very druggable target for development of cancer-associated bone disease in many settings, as well as in osteoporosis. So the argument they like to make is that the tumor-dendritic cell interaction is really a two-way street, that it leads to enhanced survival and growth and dedifferentiation of tumor cells. We have shown you that it can induce capacity for cell fusion, which can lead to osteoclast formation in myeloma. And then that these interactions now provide several very well-defined druggable targets that are potentially, many of these are now, in fact, going to the clinic to be able to be tested in the early stages of the human disease. So going back to the bedside, one of the first things that immunomodulation has taught us, and actually it continues to surprise us in many ways, is the remarkable degree to which people, even in advanced cancers, seem to respond to immunomodulation. And this is an example that was just published actually this month, or actually last month, of looking at a drug called pomalidomide, which is perhaps another analog of thalidomide, but much less anti-angiogenic compared to the parent compound, and appears to have a response. And this is just a waterfall plot showing you that the great majority of patients, in fact, do respond to this drug, even in the setting where they have received prior lenalidomide, which is the current standard of care as immunomodulatory drugs in myeloma. Now, I think I mentioned that our goal really is to move this towards trying to prevent the disease. And one of the first things you want to do in order to learn to prevent the disease is to understand the genetics and genomics of precursor lesions better. We do now know that the so-called subtypes of myeloma that we know are also there in MGUS at a genetic level. What we are trying to understand is, can we identify signatures, if you will, at the level of gene expression profiling, where a certain profile would predict for a higher or lower risk of progression by looking at the tumor cells themselves. What we are finding now is, in fact, that the microenvironment, again, at the level of GEP, is perhaps going to be more predictive than the tumor cells. Can we do immunomodulation to prevent clinical myeloma? There are two efforts ongoing. One is a study that we actually just started. In fact, Natalia just enrolled the first 2 patients this morning on that study, which takes advantage of an observation that we made some time ago, that innate immune cells are a major target of these drugs. And so combining low doses of Revlimid, which is lenalidomide, together with dendritic cell vaccine that specifically targets NKT cells as an approach to prevent clinical myeloma. The other approach that's in development and hopefully will get online in late 2010 is a national effort to compare this drug to placebo as a potential licensing trial. I think the future, however, is that it is likely that just as cancers are different between different people, preneoplastic lesions are different between different people, and we actually need to think about prevention not in the context of one size fits all, but perhaps depending on what the specific targets are for that preneoplastic lesion. And this is something I think we're moving towards, at least in terms of generating the proof of principle for that concept in humans. So I'd like to conclude that if you took nothing else from this talk, I think that the capacity of the soil, as I think as Paget put it a long time ago, to target the seed, and the capacity of the seed to modify the soil, we believe is a key determinant of the ability of the tumor to grow in the host. It's not easy for cancers to grow in people. And it really has to be the right set of circumstances, and that's really important. And that actually provides very specific targets for both therapy and actually prevention of cancer as well. So I'd like to close by acknowledging this is really a group effort, many people in the laboratory. The people who really led the work initially about the tumor DC interaction was Angelico Creja and Radek Spišek, who led the work about SOX2. This is a collaboration with many, many clinical colleagues throughout the nation. And the work with bone is a collaboration with Carl and Sagna and colleagues. Thank you very much.